All right, I think we might finally be ready to go here. All right, first 5 or 10 minutes, we're going to review some of this embryonic development stuff we already talked about, and then we're going to get into the stuff that's on your foldable assignment, and this should help finish that up. A metazoan is essentially a multicellular animal, and we're looking at how we develop as embryos. It's a 4-step process. Step 1 is fertilization, and you take a haploid sperm and a haploid egg, both with half the normal number of chromosomes. By the way, what's the symbol for haploid? N, right? Just N. And those get together and fertilize each other, and you get a zygote, a diploid, 2N. And that cell that had a lot of like mRNA that had been blocked and all that stuff goes into action, starts doing a lot of translation, protein synthesis, and it very quickly starts dividing. And that's step 2. Step 2 is cleavage. It means to split. And the cell starts to split so fast that it doesn't really grow because it's splitting so fast it doesn't have time to grow. So for the first few stages, the zygote doesn't really get any bigger. It just divides into smaller containers, smaller cells. Eventually, it reaches a stage where there's 16 cells in a ball, a solid ball. And at that point, it's called the morula. And that's spelled just like it sounds, morula. And eventually, that will catch up. That morula, though, the cells in the morula eventually make this liquid that fills in the middle of the ball, and it pushes the cells to the outside. So it hollows out the ball. Once the ball gets hollowed out, it's got a different name now. It's called the blastula. And that's kind of where step 2 ends, cleavage. Step 3, that ball of cells that we had, let's draw it up here, invaginates in on itself. Again, my picture will catch up in a minute. So if we think of this as a ball of cells with all the cells on the outside and hollowed out on the inside, it pinches in, invaginates. That opening there, that hole, is called the blastopore. And in you, what does that opening become? It becomes the anus in us. But in most metazoans, it becomes what? In most, it becomes a mouth. And this tube coming down from that hole, it's called the arc enteron, or sometimes just the primitive gut, and eventually that becomes what? What does the tube become? The digestive system. Becomes the digestive system. While this is going on, the cells that are in that ball organize themselves into 3 layers. One layer on the outside, and it's called ectoderm. One layer in the middle, called mesoderm. And one layer on the inside, called endoderm. And those 3 layers of cells are your germ cell layers. They're essentially your stem cells. And those 3 layers of germ cells are going to make you. But that process where the ball pinches in on itself and makes the blastopore, and where it organizes itself into 3 layers, that's called gastrulation. And by the time the cells are all into the 3 layers, now the ball is called the gastrula. So we got 3 different names for the ball of cells, depending on what stage it's in. When it's just a 16-cell solid ball, it's called the morula. When it hollows itself out with fluid in the middle, it's called the blastula. And then when it finally gets organized into the 3 germ layers, it's called the gastrula. And then step 4 is organogenesis. And that's where the germ cells go to work and they make your organs. That's what organogenesis means, creating organs. And one of the very first things that's made during this process is the neural tube, and that becomes what? What the neural tube become? Spinal cord and the brain. So that's one of the first things that happens. But that's most of the embryonic development steps, at least. Computers want to be a little ornery today. Another thing we talked about were protostomes versus deuterostomes. The word protostome means, proto means first, right? First, and stone means what? Means mouth. So, well, that's up there on the screen. Protostome means mouth first. These are animals where that blastopore becomes the mouth. And there's something incorrect technically on here. Really, the protostomes are these three groups of animals. The annelids. By the way, what are annelids? Things in phylum Annelida. What are we talking about? Segmented worms, like earthworms and leeches, stuff like that. And then you got mollusks, which are things like clams and oysters, snails, slugs, even like an octopus. And then finally, arthropods, which are mostly what? For arthropods, mostly insects, but also the crustaceans fall into that group. Things like crabs, lobsters, shrimp. Arthropods, Arthropoda is by far the biggest phylum, the most diverse phylum. Those are protostomes. That blastopore becomes the mouth. We'll review some other differences in a minute too. We, on the other hand, belong to a group called deuterostomes, which means mouth second. Anus first. The blastopore becomes the anus. And our group includes all of phylum Chordata, which is what we're going to focus on today. Chordata, the vertebrates, all the vertebrates. And also the echinoderms, which we said in here before are like what? What are echinoderms? Starfish and sea urchins. So according to this, other than the other vertebrates, echinoderms are essentially your closest relatives through evolution, other than the other vertebrates. So the other thing we want to review here with the protostomes and deuterostomes, big thing to know. One, in protostomes, the blastopore becomes a mouth, but the other is the way the cells divide. Protostomes have what's called determinate cleavage. It's called determinate because what's determined really early? The role of the cell. Methylation happens really early during embryonic development, and even by this 4-cell stage, when there's only 4 cells in the embryo, what's already happened? Those cells have specific roles, which means really they're no longer what? They're no longer stem cells. They're no longer, what's that fancy other word we use for stem cells? Totipotent. They're no longer totipotent. So if you were to remove one of these cells, this embryo dies. That's in protostomes. But in deuterostomes like us and other vertebrates, our cleavage is said to be indeterminate, which means our cells remain as stem cells quite a bit through the process. And if you were to take one of these 4 cells away, this keeps growing because it just replaces that cell with more cells. Because these cells are still stem cells and they haven't been methylated yet. Again, remember, protostomes are things like arthropods, insects, annelids, and mollusks. And deuterostomes are things like all the vertebrates and all the echinoderms. Now we talked about the germ layers. Remember, the germ cell layers are essentially your stem cells. There's 3 layers of them. Each layer does different things. The innermost layer of the gastrula, that ball of cells, is the endoderm. And it makes stuff mostly very deep within your body, like your digestive system, your lungs, your liver, your pancreas. The mesoderm is the middle layer of stem cells. And it makes stuff that's typically in the middle of your body, like the middle layer of your skin, the dermis, the bones, the cartilage, the blood vessels, the linings of your body cavities. And then the ectoderm is the layer on the outside. And it makes a lot of the stuff that ends up near the outer part of your body, like the epidermis. A lot of the sense organs on the outside, like your ears and your eyes, the tooth enamel. And it also makes the epithelium, or the linings of most of your body's openings, like the mouth and the nose and the anus, and so on. So again, these are the germ cell layers, which are essentially your layers of stem cells, and what they do. And then we'll end this by again mentioning that the germ cell layers are your stem cells. Remember that stem cells are cells that haven't differentiated yet, mainly because they haven't been methylated. Remember that methyl groups get added to what? The cytosines of the DNA, and they cause it to do what? Coil up real tight and essentially be off, and be off for how long? Permanently. It's a permanent way to turn off the DNA. So it's a way to essentially program cells to be a certain type of cell. There's hope that one day stem cells can be used to treat all kinds of problems in your body. It's already been used to treat leukemia, which is a problem with your bone marrow. Maybe be one day used to treat cancer, different kinds of damage in tissues, maybe to fix that damage. There's a lot of hope that it might be used especially to treat like spinal cord injuries or even brain injuries. Even diseases like Parkinson's or maybe Alzheimer's disease as well. But right now, most of that is still in its infant stages.
Dr.
Mm-hmm.
Did you see the thing about the scientists in Australia who found the Alzheimer's possible cure that they were working on?
No, I did not.
They tested like a ton of rats, like 75% of them getting their memory back.
What was he? You know what he was treating
I
them
don't
with?
remember, it was on the news the other day.
It was in Australia. I'll have to look that up. That's cool. I'll have to look that up. All right, one more thing on this PowerPoint, and then we'll switch over to the chordate stuff. Body cavities. There are a few animals that are acoelomate, which means no body cavity. How could you get by with not having a body cavity if you were an animal? You don't need a body cavity if you don't have what? If you don't have organs. There are a few real simple animals like sponges, like jellyfish, that don't really have organs, so they don't need a body cavity. There's one phylum of animals, nematodes, that have what's called a pseudocoelom. And that actually means like false coelom, right? False body cavity. It's not really a false body cavity, it's just made differently than the one that other animals have. It forms between two germ layers, the endoderm and the mesoderm. What are nematodes, by the way? Those are roundworms, like they're found in soil mostly. They can cause some parasitic diseases like pinworms and hookworms and so on. Most animals, though, have a coelom. That's just your standard body cavity. It's where your organs are at. It's a space for holding organs. It's the inner part of it is lined with cells from the middle germ layer, the mesoderm. And like I said, most animals have it. Annelids, the segmented worms, mollusks, clams, oysters, snails and slugs, arthropods or insects, echinoderms like starfish, and the vertebrates, the chordates, all have a coelom. All right. That's stuff we've been through before, so let's get out of this for a second. And then we'll talk about the stuff that you're going to see on your foldable assignment. So, this foldable assignment is about phylum Chordata. Phylum Chordata are mostly vertebrates. There are a couple of things in phylum Chordata that don't have a spinal cord, though, or not a spinal cord, a backbone. We'll talk about those in a minute. But these things that you see on the screen right now are things that make chordates be chordates. I think this is one of the things you have to put in your folder for, these characteristics of things in phylum Chordata. And one thing that all chordates have at some point in time in their life is this rod along their back, a dorsal rod, called the notochord. Now, in most chordates, the vertebrates, that notochord goes away during embryonic development, and what's left of it becomes the little disc in between your vertebrae. What are those discs made out of in between your vertebrae? Cartilage. And what's the function of them? Why would you want little spongy discs between your backbones? Yeah, to cushion it so that the bones don't rub on the bones. What happens to those discs during the course of the day? Yeah, they get compressed. So you actually get shorter during the day. Gravity shortens you up by a measurable amount. And then when you lay down and rest, they expand back out. But again, all chordates have this notochord, at least during embryonic development. There are a few that keep it their whole life. We'll talk about them in a minute. Also, all chordates at some point in time in their life have pharyngeal gill slits, which means gill slits along the throat. Your pharynx is your part of your throat. As an embryo, you had gill slits. You never had gills, but you had gill slits. Why would scientists say that you had gill slits? Why did you have gill slits as an embryo? There were no gills there, they didn't do anything, so why'd you have them? Scientists would say it's because you're related to what? Fish. That's where they would go with that. A lot of people think that there's some mutations in the genes that coded for these gill slits, and that eventually those mutations led to the development of jaws. So they think our jaws came from the genes that once caused us to have gill slits. Keep them. Also, all chordates at some point in time have a tail, post-anal tail. During embryonic development, you have a fairly long tail as an embryo. Also, all chordates have bilateral symmetry. Word bilateral, what does that mean? Bilateral. Two sides. So if we took an imaginary line and drew it right down the middle of your body, you have two sides that are essentially what? Maybe not the same, but mirror images, right? Two sides that are mirror images. Other than bilateral symmetry, what's the other kind of famous type of symmetry? Radial. And radial looks sort of like this, like a starfish, where you have a center part and pieces radiating out like spokes on a wheel. So most of our animals have bilateral symmetry. All of our chordates do. And then chordates are also deuterostomes, which means the anus forms from that blastopore, and our cleavage pattern during embryonic development is indeterminate. So these are things that make chordates be chordates. Now remember that all vertebrates are chordates. But there are some chordates that aren't vertebrates. And that's what we're going to touch on first. This thing here, a lancet. Sort of looks like a fish, maybe a little bit. It's not a fish. Right along its back, it has a notochord that it keeps its whole life. It doesn't have a backbone. It's not a vertebrate. It's got little gill slits up here in its pharynx. What this thing does, it lives in the ocean, and this back part of its body, it buries in the sand, and then this part right here sticks up, and it essentially is a filter feeder. But it's supposedly one of the very earliest chordates. It has a notochord, it has gill slits, it's a deuterostome. That's what's characterized as a chordate. Now there's another one that's even more confusing. This thing's called the tunicate. And just looking at it here, what does it look like? What does that look like? What kind of animal? It looks like a sponge. And you can see it's filtering the water like a sponge. But it's said to be really closely related to a vertebrate. Is a sponge very closely related to you? No, but this thing is. And the reason they think it is closely related to the other vertebrates is that as a larva, it looks almost just like this thing. And then it goes through almost like a metamorphosis period where it turns into this thing. But during its larval stage, which looks a lot like a lancet, it has a notochord, it has gill slits, it has all those chordate characteristics. As an adult, it loses a lot of those. It doesn't have a lot of those things as an adult. All right, those are chordates that are not vertebrates. For the rest of our time today, we're going to focus on vertebrates. So these are some characteristics of vertebrate chordates. Compared to other animals, vertebrates are large and very active. We have a high degree of what's called cephalization. What do you think that means, cephalization? Important to know those letters right there. Cephal means head. Cephalization means that most of your sense organs are concentrated in your head. So most vertebrates, sense organs, most of them are located in the head. As a vertebrate, you have an axial endoskeleton, runs up and down your body, supported by a backbone. And that skeleton grows with us, unlike what kind of skeleton? What kind of skeleton doesn't grow with the animal? What did you say?
Exoskeleton?
Yeah, an exoskeleton, which is what kind of things have? Yeah, arthropods. Yep. So that's nice. We have two pairs of appendages, arms or legs. We've got those cartilaginous discs in between our vertebrae, help to cushion. Remember, those are derived from what used to be the notochord. We have a really good respiratory system. And that really comes down to, the importance of it comes down to what, 3 letters? Why is it really important to have an advanced respiratory system? What's a 3-letter answer to that?
Air.
Nope. ATP. Because without oxygen, you can't do what? Cell respiration. And really, the next one comes down to ATP as well. Good circulatory system. Because once you breathe in that air, that oxygen, you have to get it to your tissues so they can do cell respiration and so they can make ATP. A good digestive system is very similar because you got to start with the food, right, to do cell respiration. And then in addition, as vertebrates, there's a pretty advanced nervous system to let you sense and adapt to your environment. So these are all characteristics of vertebrate chordates. Did you get what you need, Cliff? All right, we're going to try to hit maybe 2 groups of the vertebrates before we go today. This is a phylogenetic tree. It's supposed to show what? What's this thing supposed to show us? How things evolved. It's like a family tree of evolution. Down on the bottom left, what's supposed to be in the bottom left of a phylogenetic tree? The oldest things, the most primitive things. And then as we go up and to the right, you're getting the things that have evolved more recently. So mammals, the most recent, lancelets, the oldest. And we just go up this way. So this is a phylogenetic tree of chordates, phylum Chordata. What's implied to be down here at the root of that? Say again? Yeah, the common ancestor of all of those things. And whenever you get to a branch, for example, maybe you get to that branch right there. What's implied to be sitting right there at that branch? Common ancestor of amphibians and reptiles, birds, and mammals, right? So whatever this thing was, its ancestors became amphibians and all these other groups. But right here is implied to be just what? Common ancestor of reptiles and birds. So we've got our two really primitive chordate groups over here on the left, the lancelets and the tunicates. The first real group of vertebrates we're going to talk about is a group called Agnatha. And these are the jawless fish. By the way, before we move off this phylogenetic tree, what are these things written along the axis like that? Yeah, they're traits. They're called derived traits or derived characteristics that help one group evolve into the next group, right? Like adaptations that allowed that one group to descend from the group before it. So this first group of fish we're going to talk about, collectively, like I said, they're often called Agnatha. It turns out Natha or Natho means jaw. So this means no jaw. That's what the word Agnatha means. There's two kinds of these that you might hear about. These things called hagfish. Scientists think they were the first fish to evolve. They don't have jaws. They typically live on the bottom of body of water, and they filter feed or they scavenge. There's a lot of hagfish in like the Great Lakes. There was an episode of Dirty Jobs about hagfish. Apparently people eat them. They also are coated in this nasty mucus coat. That's why it was a really dirty job to catch them. They're sort of the size of a small snake. And they almost look like a small snake. We've got a picture that we'll look at in a minute. Maybe the more famous group of agnathans, though, are these fish called the lamprey. They keep their notochord, but it gets surrounded by cartilage. So they're generally considered to be the first vertebrates. Because they have a back, not really a backbone, but they have a back cartilage. They don't have any appendages, no arms or legs, but they do have a dorsal fin, like a shark. And they have a tongue that's said to be like a radula. You know what a radula is like? A snail has one of these too. If you were to blow up their tongue under a microscope and look at it real close, it looks like a chainsaw blade. What do these lampreys eat? Let me show you a picture. I'll come back to this slide. Here's a lamprey. So you can see his gill slits. That head is like a sucker. It's actually, I think it's attached right now to the camera lens, is how they got this picture. It's like a sucker, and its tongue, which you can't really see very well in here, it'll bite on to a fish, and it'll basically suck a hole in the side of the fish and then like drink its blood and its guts. It's like a vampire fish. Again, these live in the Great Lakes. There's actually one species that lives in the Flint River of lamprey. Anyway, they're the first fish, no jaws, which means what they can eat is sort of limited, right? Jaws are a big deal. If you got jaws, you can eat a lot more things than you can without them. So they're sort of limited by not having jaws. The other fish that we're going to talk about are called gnathostomes. Again, remember, this part of this word means jaws, and this part of the word means mouth. So mouth with jaws. There's this set of genes in your body that we haven't really talked much about. I don't know if we've ever mentioned those genes before. All animals have this set of genes called the Hox genes, H-O-X. And these genes are the genes that basically code for your body's shape. and structure, where things are at and how they're made. Most scientists think that there was a mutation in the genes, in these Hodge genes, that originally the genes coded for gill slits, but the mutation led to them coding for jaws. And that's where the first jawed fish came from, was due to this mutation. The first organisms on Earth, according to scientists, that had jaws are fish that belong to this group, chondrichthyes. And again, this part of this word, chondra, that means cartilage, and ichthyes means fish. Because a scientist that studies fish is an ichthyologist. These are the cartilage fish. They're sharks, and also the rays, like the stingrays and the manta rays. The only teeth in their body, the only bones in their body are their teeth. Everything else is made out of cartilage. What's the advantage of that? What's the advantage of having a cartilage skeleton instead of bone skeleton? Yeah, it's lighter and flexible, which means you probably can move faster, right? What's the disadvantage? It's easier to break. It's not as strong. The chondrichthyes, the cartilage fish, have this really thick skin. Their skin is almost toothy. You can cut yourself on a shark's skin. It's sharp. The scales are almost like teeth. But the reason they need that is they're living in the ocean, which means water's wanting to do what? If you're a fish living in the ocean, what's water wanting to do? Leave the body, right? Because water always goes from where there's less solutes to where there's more. So this is to keep them from desiccating, keep them from drying out. Thick skin protects them from that. They have really powerful fins and jaws. Good vision. And whenever you see this word olfactory or olfaction, that's about smell. You've all heard those sayings that sharks can smell a drop of blood in the water from a mile away. I don't know if that's true or not, but they can smell it really well. Most fish have a line, and if you've ever been fishing much, you notice that a lot of fish have this line that runs down their body, like where your size would be, all the way up and down. And that line is almost like their ears. It's a line that picks up vibrations and sounds. So it's how they hear and feel vibrations. And also fish and amphibians and even birds have something that you don't have. It's called the cloaca. It's at the end of the large intestine. And what it is, not only does the large intestine empty into this cloaca, but so does the bladder, and so do the reproductive organs. And then the cloaca empties out. So it's sort of the holding area for poop, for urine, and for sperm or eggs. They all come out the same place in fish, amphibians, and also birds. So those are cartilaginous fish, chondrichthyes. And that's probably about as far as we're going to go with groups. There's bull shark. What's a bull shark sort of famous for? Yeah, well, it'll attack people. It's one of the main sharks that bite people. But what else is it famous for? Yeah, it can go into fresh water. There have been bull sharks found near St. Louis. How far is St. Louis from the ocean? A long, long way. Hundreds of miles. They can live in fresh water for a long, long time. And actually, they usually give birth in fresh water. Why would you want to do that if you were a fish, a shark? Why would you want to go in the river and have your babies? Not really about food, I don't think. There's not very many big predators in the river, right? The number of big predators in the river is much smaller than there are big predators in the ocean. All right, so we'll end with this today. So we talked about the lancets and the tunicates. And the thing that separates them from the agnathans, we could write it right in here, is the vertebrae, the backbone. And then we've got the jawless fish, like the lampreys and the hagfish. the thing that separates them, the derived trait that separates them from the cartilaginous fish like sharks, is the development of jaws. And then where we'll go next time is we'll talk about the bony fish, which are most fish. And notice it says lungs. Do most fish have lungs? No. But there are a few that do. Where would you find a fish with lungs? I've not ever heard of this before. There are several species of fish in Africa that live in streams. And the streams dry up when it doesn't rain for long periods of time. So what do the fish do? What do fish do when the water's gone? They dig a hole in dirt and they breathe air until it rains again. Those are lungfish. We'll talk about them, but they think that these lungfish are the ancestors of the first land animals, which are amphibians. And we'll get to that the next time around. That's a good place to stop for right now.